So drugs often act at neurotransmitter receptors, right? And chronic drug use can change, that receptor distribution, number, etc. It can upregulate the expression of specific receptors. It can downregulate the expression of certain receptors. And several techniques allow us to sort of track changes in receptor distribution, number, how much affinity they have for, specific drugs, etc. So these are all techniques that are designed to sort of help you visualize the number, the type, and the distribution of various receptor targets, for example, in the brain. And one of them is something called radioligand binding, right? So a radioligand is, it's the name of it tells you what it is. Radio refers to, radioactive. So this is something, some kind of a radio label, something that's going to decay and then thus be measurable, be visualizable, right? If that's a word. And then ligand is chemical, right? That's a fancy word for chemical. So we're adding a radio label, some kind of a radioactive tag, something that'll decay to a chemical. Let's say a drug that you want to see where it acts in the brain, or say it's some kind of a chemical that will bind to, for example, the D2 dopamine receptor, and you want to, introduce it and then, count either in a living animal. You can use techniques known, the one's called Positron Emission Tomography, or PET, which can, record the decay events and, sort of spatially identify where they're coming from to look for changes, let's say, in D2 receptor expression. Let's say you have, an animal that you're giving cocaine to over 2 weeks, and you're going to hypothesize that there's a reduction in the D2 receptor, expression. Well, you can actually, basically administer this radio label and then, sacrifice the animal, and we're going to do an in vitro experiment, right? And then grind up the nucleus accumbens and then, measure the amount of, radio labeling, decay that we've got in that particular, experimental group versus like a control group that never got, any cocaine over 2 weeks. And, there's another important aspect of this, this technology. You can actually do what's called receptor autoradiography, which is graphing, basically sort of like viewing the distribution of where these radio-labeled chemicals are binding, presumably to receptors in this example, across like a slice of brain. So you could see like where is the receptor distribution, in a sort of a graph, right? It's sort of a landscape across the brain. And I've, the example here is, it turns out that those who routinely, smoke cigarettes, they get lots of nicotine exposure. Well, that nicotine acts at nicotinic cholinergic receptors. Initially, it stimulates them. It acts as an agonist, but then they desensitize, as nicotinic cholinergic receptors will do. They'll switch into a form where they're not paying attention, and that will lead to changes, a drop in this biochemical cascade, or a change in this biochemical cascade inside these neurons back to the genes, and then you'll get changes in expression, typically significant upregulation, many more nicotinic cholinergic receptors in the brains of smokers. And this is, a paper here and an image that shows you, there's a lot more binding, of these radio-labeled chemicals in the cortex, and in specific layers of cortex in the brains of smokers versus non-smokers. So this radioligand binding and this, and receptor autoradiography can tell you important information about, the distribution, the subtype, the level of expression of these different receptors in response to, drugs.